Hello and welcome to TYBSc students in this second lecture about the topic linkage and recombination. In previous lecture we have studied about the definition or different definitions of linkage and different views about this particular concept by Thomas Morgan and Bateson and Punnett. In today's lecture, we are going to study about the salient features of the theory of the linkage and different examples about this particular linkage. Now we turn towards the salient features about this particular theory of linkage. Morgan and Castle formulated the chromosome theory of linkage. So, to understand this particular theory of linkage, we must know about the particular concept of gene or the definition of gene, which we have studied in the first unit, that is Mendelian genetics, in which we have seen this particular concept of gene, which I have shown in this particular picture, and the gene that show linkage are situated in the same chromosome. So this is the particular segment or DNA fragment which is called gene. Genes are arranged in a linear fashion in the chromosome. The genes which are closely related show strong linkage, whereas those which are widely separated have more chance to get separated by crossing over or they having weak linkage. Linked genes remain in their original combination during course of the inheritance. Now we will see the particular in the theory of linkage, the genes which are linked having specific type of arrangement. The linked genes show two types of arrangement on the chromosome. If the dominant alleles of the two or more pairs of the linked gene are present on one chromosome and their recessive alleles of all of them on the other homologue, that is capital A, capital B, small a, small b. This particular arrangement, this particular which is shown in this particular picture, that capital A, capital B and small a, small b, this particular arrangement, this type of particular arrangement is called cis arrangement. However, if the dominant allele of one pair and recessive allele of second pair are present on one chromosome and recessive and dominant allele on the other chromosome of homologous pair, that is capital A and small b and small a and capital B. This particular arrangement is called trans arrangement. Now we will see the different examples about this concept of linkage. So the first example in this particular concept of linkage is about the maize. So Hutchinson crossed a variety of maize having colored and full seed, which is having genotype capital C, capital C, capital S, capital S, with a variety having colorless shrunken seeds, that is small c, small c, small s, small s. in this particular diagram we will see this particular colored full seed. This is the particular colored full seed and colorless shrunken seed, which is having the particular genotype. The gene C for color is dominant over colorless allele. Capital C is dominant over the small c, which is having colored gene, which is dominant over colorless, and the gene S, capital S for full seed is dominant over its shrunken allele, that is capital S is dominant over small s. All the F1 plants produced colored and full seed, but in a test cross, when such F1 female heterozygous are cross-pollinated with the pollen from a plant having colorless and shrunken seeds, that is double recessive, then 4 types of seeds are produced. This particular diagram shows 4 types of seeds. From the above stated result, it is clear that the parental combinations are more numerous, that is 96%, 96.4%. This is the particular parental combination having percent 96.4. Then the new combination. This is the new combination. These are the new combinations. This particular combination shows having percent 3.6. This clearly indicate that parental characters are linked together. Their genes are located in the same chromosome and only in 3.6% individuals the genes are separated by crossing over. This is an example of incomplete linkage. Second example about this particular concept of linkage is about the Drosophila melanogaster. Morgan, that is Thomas Morgan, in 1911 crossed an ordinary wild type of Drosophila with grey body and long wings, that is having genotypes capital B, capital B, capital V, capital V, with another Drosophila which is mutant type with phenotype black body and vestigial wings. Vestigial means remaining in the form of signs. Vestigial wings having genotype capital B, small v, small v. All the hybrids in F1 generation are with grey body and long wings having heterozygous genotype capital B, small b, v, capital B, small b, capital V, small v, that is phenotypically like the wild type of parents, that is grey body and long wings. If now a male of F1 generation capital B, small b, capital V, small v is back crossed with the double recessive female which is test cross having black body and vestigial wings, only parental combinations are formed in the F2 generation without appearance of any new combination. The results indicate that grey body character is inherited together with long wings. It implies that these genes are linked together. Similarly, black body character is associated with the vestigial wings. Since only parental combinations of character appear in the offspring of F2 generation and no new or non-parental combinations appear. This shows complete linkage. The complete linkage is seen in Drosophila melanogasters males. Now we will go quite deep in this particular theory of linkage. For that purpose we have to understand about the linkage group. Here I have shown these particular pictures about gene which I have repeatedly showing in these particular slides for your understanding that how gene is present on the chromosome. Genes are located in same chromosome, are linked to it and inherited as a single group. Thus each chromosome is linkage group. There are as many linkage groups as there are pairs of the chromosomes. For example, 4 for the Drosophila melanogaster and 23 for the man, 10 for the maize and so on. Since the chromosomes ordinarily duplicate at the time of gamete formation, all the genes present on a particular chromosome will be transmitted to get to the gamete as a single block or unit. The linkage is not due to any relation between two genes but is simply because they happen to be located in the same chromosome. Experimentally, it has been found that linked genes are not always inherited as a single unit, but nearly they tend to remain together. It has been also been observed experimentally that larger the chromosome, larger will be the number of genes on them. Small chromosomes have got small number of the genes. Thus, in Drosophila, there are 4 linkage groups corresponding to the 4 chromosomes. Each linkage group contains many genes responsible for the characters. 2 linkage groups in Drosophila are exceptionally long. One is too small and another one is intermediate in the size. So these are all 4. In maize there are 10 linkage groups. In barley 7 linkage groups. In cereal plants and animals all the linkage groups have not been discovered. For example, in mouse 16 linkage groups for 20 pairs of the chromosomes and in rabbit 11 linkage groups for 12 pairs of the chromosomes are known so far.